Okay, so actually this lecture has been partially also developed by Professor Anna Orlova, who is a course leader for this particular course, and she is also a professor in molecular imaging at Uppsala University. And before we start, I would just like to give you a short reminder. I'm sure that, I guess your background is pharmacy, right?
Yeah, for me, I'm done with my pharmacy course. I'm doing masters in drug discovery and development.
Okay. And for Guevara?
Yeah, I'm pharmacy as well.
Okay, good. So I'm pretty sure that you know some things that we will discuss at this lecture, but I just want to give you a short description of what a peptide is and how it differs from a protein. So both peptides and proteins, they consist of amino acids, but peptides are shorter fragments. So they consist of 50 or less amino acids. And if we think that molecular weight of one amino acid is about 100 Dalton, then the molecular weight of a peptide is generally below 5 kilodalton. So everything that is bigger than 5 kilodalton or that consists of more than 50 amino acids can be considered a protein. I guess you can see the screen that I share, right? The presentation.
Yes.
Okay, good. So when we talk about positron emission tomography, we know that this imaging modality has really a lot of advantages. So it has a high sensitivity, high resolution, which means that we can distinguish quite well different parts of the image on an image. And then it also allows to do quantification of the signal. So these are the advantages. The disadvantages of this method could be a short half-life of positron emitting radioisotopes. So it's not very suitable for kinetics of large proteins such as monoclonal antibodies, but it still can be used, and we will look at some examples. And the third limitation of this method is that these radioisotopes that emit positrons generally mainly produced by cyclotron. We will also talk later about that. So when it comes to the radioisotopes for PET imaging, there is really a large number of isotopes, but how do we select the ones that we can use? And that there are several categories or several things that determine our choice. So basically the first thing we should consider is the half-life of the radioisotope. There are isotopes with a short half-life and with a long half-life. And depending on what we want to study and depending on our molecule of interest, we can either choose a long-lived or a short-lived radionuclide. The second category or the second characteristic is availability of the nuclide. Sometimes we are limited by what is currently available, what can be produced. For example, if the hospital, if there is a cyclotron, can they produce this radionuclide at the site, or we have to buy it from somewhere? Sometimes it can also be the price. For example, SPECT or SPECT is a very common imaging modality in most of the world and in most hospitals in the world, especially outside Europe. SPECT is widely used, and one of the common nuclides for SPECT is technetium-99m. That technetium-99m is widely available because it can be eluted from a generator. So generator is a small portable device or instrument that you can transport. So every hospital can buy this generator, install, and just elute technetium for around a week. So that ensures that they will always have the radionuclide at the site. But for PET, currently there are only generators for gallium. So that limits the choice of radionuclides that we can use. A third characteristic that can affect the choice is the image quality of PET, and we will also look into that. Not all nuclides give the same, have the characteristics that would result in the same image quality. For some nuclides, image quality is better, and for some is worse. So the fourth characteristic is the labeling method. Of course, we have to look at the properties of the radionuclide, at the nature, and then we will be able to see which labeling method we can use to attach them as labels to our targeting molecule, to our protein or peptide. Another question that can affect the choice is, what do we want to study? Do we want to study the pharmacokinetics of a protein? For example, if it's a long-term pharmacokinetics, we want to have long-lived nuclei that would allow us to study the kinetics at late time points. Or if we want to just do imaging, for example, we are developing imaging probes, and then probably we'll do not need a very long-lived radionuclide. And the last point that can also influence the choice of the isotope is influence of labeling method on studied protein. Excuse me, sorry. Yes, another point can be influence of labeling method on studied protein. And we will look into details how the labeling method affects the biodistribution of the protein. So we can see that on this slide, we can see the characteristics of various targeting molecules that we can use in our research. For example, we can see a large variety of proteins and peptides and different and their comparative sizes. And we can look at the left side, we have a monoclonal antibody, which is about 150 kilodalton size. And we have the antibody fragments that are a lot smaller in size. And we have smaller targeting molecules that we work in our lab with. They're called affibodies. Maybe you have heard about them before. So affibodies are about 6 kilodalton in size, and then there are also shorter peptides, such as about 1.5 kilodalton, and then of course there are small organic molecules less than 1 kilodalton that could be also used for targeting. So what could be the difference? Well, when it comes to behavior in the body, we have to look at blood clearance rate. Blood clearance rate is basically the rate at which these molecules are cleared from blood, how fast they're cleared from blood. So the large molecules such as monoclonal antibodies stay in the blood for a very long time, and for some monoclonal antibodies the half-life in blood is weeks, sometimes 1 week or 2 weeks. And the smaller the size, the faster these molecules are cleared from blood, and also the smaller the size, the better they can go into the tissue, penetrate into the tissue and reach tumor, for example, if we're interested in imaging tumors. And then we have to remember about several biological parameters. One parameter is called kidney cutoff value. So have you heard about this before, about the kidney cutoff?
No.
For a glomerular filtration barrier, maybe?
Oh, yeah. Yes.
So it's the same. It's the same, just a different terminology. So a glomerular filtration barrier is approximately at 60 kilodalton, which is basically determined by the hydrodynamic size of the molecule, and that about 60 kilodalton molecules that are bigger cannot be filtrated through the glomeruli in kidneys, and they will stay in the blood longer. But molecules that are smaller than 60 kilodalton, approximately, they can be filtered in the primary urine, and removed from the body through kidneys, through renal excretion. And then there is another parameter which is called the EPR effect. Have you heard about it before?
Yeah, I think so.
No, okay. So it's something that is related to tumors in cancer research, and the EPR effect is considerable at about 45 kilodalton. So EPR is enhanced permeability and retention effect. What it means is that when tumors grow, they usually grow very, very fast. And the vasculature doesn't have time to grow properly. So the vasculature in the tumor has a lot of abnormalities, a lot of gaps in between the healthy endothelia. So the vasculature is very leaky, which means that targeting molecules such as large monoclonal antibodies, which are bigger than 45 kilodalton, can penetrate through the gaps in the vasculature, and they can accumulate in tumor tissue. But that is basically non-specific accumulation. So it's not the accumulation is not mediated by interaction of the targeting molecule and the target, or like a receptor on cell surface, but it's just passive targeting by the so-called EPR effect. So when we talk about if we look at all the nuclides that can emit positrons and can be used for PET, this is basically the table that summarizes the nuclides, their half-life in hours, and their reactions used for production. And we can really see that indeed we have quite a large choice of nuclides, but then we can categorize them into different categories. So first is availability of the nuclides. So most of them have to be produced using cyclotrons, and cyclotrons are quite expensive to manage. You need to have quite good logistics and qualified staff, which is not available in every hospital in the world, so that puts a limitation. The use of enriched targets and the necessity to regenerate target material, it simply means that the cost of such production will be very, very high, which is also limits the basically the availability of the nuclides. And currently, the ones that you can see on this slide, the nuclides in black, are all produced by the cyclotron, and only two nuclides, scandium-44 and gallium-68, can be produced by generator. And as I mentioned before, generator allows us to produce the radionuclide at the hospital site. It's a very small portable device, and it can be bought. For example, gallium generator can be bought and can produce gallium for 1 year. It's quite easy to use, and we also use it in the lab. So really the availability is puts quite a big strain on what we can use. What concerns scandium-44, there are only two generators in the world, as far as I know, right now, so it's just limited as well. So basically, the only choice of gallium-68, and why gallium-68 is so popular, is because it can be produced by a generator. And now we can look at other nuclides that can be produced on a cyclotron. And the first that we can see in red are the nuclides oxygen-15 and nitrogen-13. They have a very, very short half-life of just several minutes. So it's not so useful for labeling, because labeling of proteins is a chemical reaction which takes a certain amount of time. Then if you need to do purification, perhaps you need to do quality control. So if the half-life of your nuclide is just minutes, it's very inconvenient to work with. So oxygen-15, for example, is used as radioactive water, and is actually used in Uppsala University Hospital as well. It's used as a tracer for measuring blood perfusion, for example, for measuring heart function, but it's not, oxygen-15 is not used for labeling of biological molecules. Then we have a second group of nuclides, which are highlighted in red here, is vanadium, manganese, and forgot the English name of this element. Basically, the ones that you see in red are nuclides which have isotopes which have extremely long half-life. So they are not so useful because when we inject something in patients, we don't want the activity to stay for too long in the patient because it will give a high radiation dose. So these are not used for labeling of biomolecules as well. And then we have the rest. The rest here, which basically in black in red we have the shortest half-life, less than 2 hours. So carbon-11, fluorine-18, gallium-68 are the most commonly used ones with a half-life of less than 2 hours. Then in black we have all the other isotopes with half-life of from 3 hours to 1 day. And in the blue we have only zirconium-89 and iodine-124, which have a half-life of over several days. So why is it important to know the half-life of the radioisotope, and why is it important to know the size? Because basically it's good to match the size of the targeting protein with the half-life of your isotope, because big molecules have slow kinetics, so they will circulate in the body for a long time. So if you want to detect, or if you want to study them, if you want to detect some biological processes, we should also use nuclides with long half-life that would allow that. For example, for antibodies, it will be nuclides that are here in blue. So it will be zirconium and iodine because they have the longest half-lives. And for the smaller molecules or intermediate size, we can use the radionuclides that have as well short half-life. That would be a good match with the fast kinetics of the small molecules. So isotopes with long half-life are suitable for labeling of large biomolecules, and isotopes with short half-life more suitable for labeling of small biomolecules. And then we can have two kinds of situations what we want to study. On this graph you can see, for example, a biodistribution of a targeted protein over time, and here at the initial time point we have, for example, a time point of administration or injection. After some time there will be maximum accumulation in the tumor, and with the longer the time, there will be excretion from the body and loss of the signal. So if what we want to study absolute accumulation, for example, on the maximum point, then we can use short-lived isotope with a half-life that would allow us to do that, and the half-life is approximately, can be basically, if you want to observe something for some period of time, your half-life can be up to 3 times of that period. So if your half-life is 1 hour, you can observe something at 3 hours post-injection. For example, for gallium-68, the half-life is 68 minutes or around 1 hour. It means that if we label something with gallium-68, we inject it to patients, we can observe the processes, we can do imaging up to 3 or 4 hours post-injection. After that, we already will lose too much signal and we will not be able to detect anything. But the second situation is when we want to do experiments at later time points after injection. For example, we want to study some processes up to 7 days post-injection, which means that we have to take a nuclide, an isotope that would have a longer half-life that would allow us to do that, to do late time point imaging. So the choice, as I said before, really depends on your application, what you want to do. Do you want to do imaging at short time point, or you want to do you want to look at the metabolic status, at the washout from the tissues, and so on. So in case 1, we are interested in absolute accumulation, and in case 2, we look at the washout, for example, or clearance. And on this slide, you can just see a summary, basically a matching between the half-life of the radioisotope that can match different sizes of targeting molecules. So for short peptides that have the shortest half-life, biological half-life in the blood, we can use gallium-68, for example, or fluorine-18. For intermediate size scaffold proteins, like affibodies, which are between 5 kilodalton to 20 kilodalton, and so on. There are some other options with the intermediate half-life, and we can also use the shorter lived nuclides, but we cannot study processes longer than, let's say, 3-4 hours post-injection. And then for the monoclonal antibodies, we cannot use the shorter lived ones, so we can only use the longer lived ones. And another level of complexity that will now come into question is the nature of these radioisotopes. Some of them are metals, so they have a characteristic of radiometals. Some of them are halogens. And if you remember from the periodic table, halogens are located at the right side of the periodic table in the seventh group of elements. So there we have chlorine, fluorine, iodine, bromine, and astatine. So in the darker gray, we can see here radiohalogens such as iodine, bromine, fluorine, and the labeling chemistry for them is totally different from radiometal chemistry. And the lecture that will be after that will be given by Bo, who is also a postdoctoral researcher, and he has a large experience working with halogenation, especially with fluorination, so that he will talk more about that. Today, at this lecture, we will mainly discuss labeling with radiometals. So on this slide, you can see once again the same table as we have seen before, but right now we have different colors for every type of labeling chemistry that we can use. So you can really see that even if it's as you can think, it's just radiolabeling is the same, but in reality, it's actually not the same for every kind of radionuclide. The chemistry is its own, it's different, and that means that usually radiochemists are specialized in one or several types of chemistries. For example, there are chemists who only do F18 chemistry and chemists who only do radiometal chemistry, or mainly radiometal chemistry. It just depends on the on what you're working with, on your competence, on the competence of the personnel. So it can require quite different qualifications. And as I mentioned before, this table just summarizes basically optimal imaging time if you want to do imaging, at which time point we want to do imaging, then which label would be suited with a shorter half-life or longer half-life. And here we are looking again at something called non-residualizing or residualizing labels. So it's a very specific term that you probably have not heard before, and it's related basically to the properties. It's just another way of saying retained in the body or not retained in the body. So non-residualizing labels, they retained in the body less, and residualizing labels, they retain in the body more. So if you look at the non-residualizing labels, you will see that it's carbon, fluorine, bromine, iodine. So fluorine, bromine, and iodine are halogens, and it basically means that you can say that halogens are non-residualizing labels. And if you look at residualizing labels, it's gallium, copper, cobalt, zirconium, basically radiometals. So in your for yourself, you can just memorize that residualizing labels are radiometals and non-residualizing labels are halogens as well as carbon-11. So this is like the general rule. And actually, we will look a bit more into biological behavior later, and I will explain once again what is non-residualizing, what is residualizing, and why it is like that. And so we will really look at a lot of examples later on in the lecture about that. So now we can look at something that I also briefly said before about PET image quality. Basically, not every radionuclide gives images of the same quality, and it has something to do with the physical decay, with the radioactive decay that happens and for every radionuclide that decays and that emits a positron, this energy of positron is different. And the energy, when the energy is different, that means that our positron will travel in the tissue for a certain distance. So our positron travels in the tissue for a certain distance, meets an electron, and then it emits a gamma quanta of 511 kiloelectron volts into gamma quanta that can be detected by the PET detector. So if we go back a little bit, I said that different nuclides have different energy of the positron. If the energy of the positron is high, it will travel very, very far into the tissue until it meets an electron. But if the energy is low, it would not travel so far into the tissue. It will travel just a little bit and then produce a signal. So that means that for some of the radioisotopes, the image quality is much better than for the others. So on this picture, you can see, for example, for fluorine-18, the image quality is much better than for gallium-68 because positrons of fluorine-18 have much lower energy. So they are not able to travel so far. And that means that the signal that is not blurred, basically it's the ability of the PET detector to distinguish between two points that are close to each other. And if the energy of the positron is low, it doesn't travel too far, and that means that our point of decay and point of signal production are very close to each other. So we're able to distinguish each individual signal, and in this case it's just balls filled with a liquid that contains, with a solution that contains fluorine-18, and we can see that with fluorine-18 we're really able to distinguish each individual ball that is filled with the solution of the radionuclide. So for fluorine-18 is really the lowest energy of the positron. So positron energy determines the distance from decay to annihilation. And for gallium-68, for example, it has a really huge energy of the positron. So the positron just shoots out from the point where it was decayed. So it travels very far, and only then it produces a signal that can be detected. So if the decay happened here, but the signal is produced only here, you will get something that looks like a blurred image. So I just want to ask you to get some of your feedback, if you think that was clear, if you understood that part.
Yes, it was clear for me.
Okay, good. Then we could go to the next step. And basically, it's also another example that illustrates why it is important. It's important when we do imaging and when we have, for example, fluorine 18 labeled molecule. In this case, it's an aphid body, and we have a gallium 68 labeled a molecule that targets tumors. We can see here at the right upper side there is a tumor that can be visualized. And then we also have a high uptake in kidneys because this molecule is excreted by kidneys. And we can see that it's exactly the same molecule, but because of the different properties of the nuclides, the images that are produced look different. So the kidneys for fluorine 18 are very sharp and very well distinguished from each other. But on the image with gallium 68, we see that the kidneys look very blurred and very big. So it's not related to the kidneys themselves. It's not related to the properties of the molecule, biomolecule itself. It's related to the characteristics of the positron energy of the radionuclide. So it's for gallium, positron energy is very high, so we get blurred images. And for fluorine 18, we get much sharper images. Okay, so another characteristic that we can look at is abundance of positron emission. What it means is how much positrons are emitted or are produced by one decay. So we can see that for carbon-11, each decay produces a positron. So the abundance of positron emission is 100, which is very good. That means that basically we don't need to administer much activity to the patient because every decay will produce a positron that will give us a signal. In case of fluorine 18 is also very similar. So for 97% of decays, there will be positrons. And that means that we also don't need to administer high dose to get a signal, to get a good image. But in case of iodine 124, there are only 23% of positrons that are emitted. So if we want to get the same image quality as for carbon-11 and F18, we have to administer 5 times more activity because it's 5 times less in positrons that are emitted that would give us a signal eventually. So this is just something that also should be considered. Then we have talked about the positron energy, and a third parameter that is also related to the physical properties of the radionuclide is called non-annihilation gamma. It's gamma that is produced during the decay, during the radioactive decay of the nuclide. And we have this value between 350 and 650 kilo-electron volt. It's a registration window in a PET detector. So a PET detector registers everything between 350 and 650 keV. Can you imagine why it's these values? Can you think about the reason why these values were selected?
Most likely because most of them are between this range, maybe?
Yeah. Do you remember the energy of the gamma quanta that are emitted?
511.
Exactly. So it's 511 is just in between those values and then a little bit more. So then we can have a situation, for example, for yttrium-90, it naturally emits also 443 keV, 628, and 646. It just occurs together with the positron decay. There is also gamma, and it's basically your background that will give you background in the images. So for some nuclides that have this non-annihilation gamma, it just means there will be some level of background. So this is another characteristic that is individual for every nuclide and should be could be considered as well. And then there are also we should also look at some other undesirable types of radiation. If we're talking about patients, there are some kinds of radiation that are not so good for patients, that can irradiate and really produce damage to tissues. And one of these radionuclides is copper-64. So copper-64 is actually very, very popular in US, especially because it can be produced on a cyclotron, and it's popular to use in clinics, but it's not so good because only 20% of decays produce positrons. Or the abundance of positron emission is just 18%. So you need to give quite a lot of activity, high dose, and as well there is a beta minus that is produced, and beta minus is beta particles can damage DNA in cells, so that can produce damage to healthy tissues and organs. So presence of other types of radiation should also be considered when you choose a radionuclide for labeling. There is also another point that is related to basically this non-annihilation gamma, why it gives big background. So if we think about our radionuclide that decays and that produces some gamma, what the detector is measuring, it's measuring the when two simultaneous quanta hit the detector. So it measures the two events that occur simultaneously. And if there was some gamma emitted, the detector can place the source of emission in between those registration points, and that would give you background signal. So this is why if we have a nuclide that has some non-annihilation gamma, it can produce background in the detectors. So the detector may misread the signal and may place it not at the actual source, but it can place it somewhere else because those gammas can hit the detector at the same time. So this is your source of error. Okay, so if we think about applications of radiolabeled proteins, we can basically divide it into 4 different groups. The first application is in basic research. That's what we do in the lab, we do in the university. If someone wants to study some basic biological processes, they can label some protein of interest and they can study a mechanism of the disease, for example, or something else related to basic biology. A second category is the preclinical development. That's what we also do in the lab here. We work with the development of tracers for imaging of cancer, and in preclinical models. That means that we work in test tubes, we work with cancer cells, and we also work with animals, with mice that have models of cancer. So what we can do using radiolabeled proteins, we can do pharmacokinetic studies, which means that we can study the biodistribution, absorption, metabolism, excretion. We can trace the molecule using radioactivity. We can study targeting properties. how good is this molecule taken up by tumor, how long it stays there, how long it can deliver its effect, if it's a cytotoxic effect, for example. We can also do selection of doses. We can inject, for example, various amount of doses and see which dose produces the best therapeutic effect, or this kind of dose finding studies. Third group will be for clinical diagnosis. Of course, we can use radiolabeled protein for identification of patients that have a certain type of tumor. We can select those patients into some categories. For example, this patient has this tumor type. That means we can administer this type of therapy for this patient. And it's really something that has emerged in clinics or in the last 5 to 10 years. It's called personalized approach to medicine, where we are not treating all patients in the same way, but we are actually doing some stratification where we are categorizing, okay, this patient has this molecular subtype. That means that it can be treated with this kind of drug. And this patient doesn't have it, which means that we don't need to give this drug for the patient because it will not work. We can also do something called therapy response, where we will look at, for example, uptake of the tumor before treatment, uptake in the tumor after treatment, and we can see if it has changed, if the tumor size has changed, if there was a response to therapy or it was no response in progression. And we can also do radionuclide therapy. It's basically a field that is very closely related to imaging, but instead of the imaging isotopes or nuclides, we use therapeutic nuclides. So it's something called theranostics, where you can actually combine therapy and diagnostics. So for some nuclides, it's a very good match. They're called theranostic pair. Maybe you have heard, like there is yttrium-86, which is PET-emitting radionuclide for imaging, and there is yttrium-90, which is a radionuclide for therapy. So yttrium, basically 86 or 90, as yttrium has the same, it is the same radionuclide, but two different isotopes. Their labeling chemistry, labeling and chemical properties are the same, so we can use the same chemistry for both of them. We can just by exchanging the isotope, we can do either imaging, and then we can treat the same patient with the radionuclide therapy. So it's a very interesting approach that has been really working quite well. For example, for therapy of neuroendocrine tumors, maybe you have heard about neuroendocrine tumors and some other statin analogs like dotatate, lutetium dotatate. Have you heard about it?
No, not really.
Okay, basically it's just it's another area of cancer diagnosis, and for some types of cancer, which are called neuroendocrine tumors, in the recent years there has been a radionuclide-based treatment and radionuclide-based imaging that has been really used in the clinic and been really working quite well. And the second application was also for imaging and diagnosis of prostate cancer. So by doing imaging, we can quantify the signal in the image, and we can quantify how much dose will be deposited in every organ. So how much dose will be in kidneys, for example, how much dose will be in tumor, and how much will be in blood. And we can do dosimetry estimation, so we can calculate what will be the dose to normal organs and what will be the dose to the tumor. So why is it important to know the dose for therapy? Because we don't want to administer too much activity, and there are organs and systems in the body that are very sensitive to radiation. For example, bone marrow is very sensitive to radiation, and kidneys are also, of course, every organ can be sensitive to radiation, but some organs are basically very sensitive.
Is bone marrow diagnosed then if it's so sensitive to radioactivity?
Sorry, can you repeat the beginning?
How is bone marrow cancer diagnosed if it's so sensitive to radioactivity already?
I think the diagnosis for bone marrow cancer is probably some other symptoms, like symptomatic disease, like
Okay.
maybe counting of some special cells in blood, doing blood analysis. So I guess the cancer of bone marrow is like a myeloma types of
Okay.
cancer, which will produce symptoms much like characteristic symptoms. It's not a solid tumor, it's not like a tumor chunk in the body. I think it's so-called liquid cancers maybe. I'm actually not a big specialist, but you cannot do imaging to diagnose this kind of cancers. But if it's a solid cancer, if it's a tumor mass, then we can estimate how much is taken up there and how much dose we can give to the patient.
Okay, thank you.
Okay, and now we can actually go to the topic of the lecture, is actually labeling of the proteins themselves, so labeling procedures. So what we should think about when we do labeling. So imagine that we are attaching a radioactive label to our biomolecule of interest. Of course, we want this label to be stably bound to the biomolecule. It should not go away from the biomolecule, so it should be very stable. And the first characteristic is stability of tracer in blood. So what can happen in blood is that in blood we have a lot of other biomolecules that can, for example, do transchylation. If we're talking about gallium, it's a metal and it can be transchylated to ferritin. Ferritin is a large protein that is in our blood that can basically take up gallium if it's not stably bound. And then if there will be some free radionuclides like radiometal, it can also form colloids. And these radiocolloids will accumulate in the spleen and in liver. And what you will see at the end is very, very high uptake in spleen, very high uptake in liver. What it means, it can be instability of your radiolabel in blood. So it means that your radiolabel just either breaks down, the bond breaks down, or it just diffuses out of the chelator, or some other mechanisms happen. So in the lab, the first thing we do is, of course, we do the radiolabeling itself, and then we evaluate how stable is the attachment of the label to the protein. A second characteristic is a chemical modification that we introduce to the protein. Of course, when we have a biological protein, we attach a label. It is a chemical modification that can affect the property of the biomolecule. So we have to consider, for example, a conformation of the biomolecule if it changes in some way. If there was a binding site, and the binding site that the biomolecule binds to the target, and we all of a sudden block the binding site for some reason, then the biomolecule cannot bind to the target anymore. So the target recognition is somehow impaired, it's not working anymore. Something that we also check in the lab. And then there is a factor of molecular charge. So a molecular charge is a charge of the chelator metal complex. And sometimes what we generally see in the lab is if charge is positive, it's not good because it means it's high unspecific uptake in liver, in normal organs, so we will see high background. But if the charge is negative, it will be hydrophilic properties and less uptake in normal organs and less background. And of course, there is also the lipophilicity, hydrophilicity profile. We don't want to give modifications that will be too lipophilic, because lipophilic things get stuck in liver, and you will see that on imaging. If you have a chelator that is very lipophilic, you will see it in imaging. Your imaging probe, your tracer will not be good. There is a third characteristic, which is cellular processing by cancer cells of your radiolabeled protein, and it's determined by the residualizing properties of catabolites. Maybe you remember a couple slides before I said that residualizing labels are radiometals and non-residualizing labels are halogens. So later on, we will look at the cellular processing at these characteristics. And there is another parameter, which is retention in healthy tissues. Once again, it determines by the residualizing properties. So a very common target for targeting is receptor tyrosine kinases for cancer. Basically, it's what we work in the lab, but also quite a lot of other people work. And for example, it's these tyrosine kinases are expressed on the surface of the cell, so they are transmembrane receptors, and they can be targeted by some biomolecules because they are on the cell surface. So our targeting molecule doesn't have to go inside the cell through the cell membrane. It just has to react with the receptor on the cell surface and bind to it. And so for therapy of cancer, there are some monoclonal antibodies such as bevacizumab that targets VEGF receptor on some cancer cells, and there are also monoclonal antibodies such as cetuximab that targets EGFR, and trastuzumab that targets HER2. HER2 is a receptor overexpressed in some kinds of breast cancer. So these monoclonal antibodies are used for therapy in clinics, but if we radiolabel them, we can use imaging to trace, for example, how many receptors does the patient have. Only patients with a high number of these receptors can be given treatment. So basically, these receptor tyrosine kinases, they are targets for anti-cancer drugs. And we can also use them as targets for our imaging probes. And this is what we work mainly in the lab. So you will see several examples on the slides of imaging of different HER receptor families, such as human and epidermal growth factor receptor 2, and so on. Okay, so what happens after our radiolabeled protein binds to the receptor on the cell surface? So then our radiolabeled protein goes inside the cell because there is a process of internalization. So generally for receptors, they undergo internalization, and this complex of receptor, protein, and radiolabel is inside the cell and is surrounded by a vesicle, which is called an endosome, which is then converted to a lysosome. And maybe you remember that in the lysosomes, the environment is quite aggressive, so it's acidic pH, and there are enzymes that can basically destroy our protein, and it can chop up the protein into the small fragments. So some of these fragments will still carry the radioactive label, and depending on the properties of the label, depending on the nature, if it's a halogen or a metal, it will either diffuse outside of the cell or it will stay inside the cell. So the labels that stay inside the cell, they're not able to go through the cellular membranes because cellular membranes are made of phospholipids, so they are lipophilic, but radiometals and chelators, they have usually hydrophilic complexes, or they are polar. They are not able to diffuse outside of the cell. That's why they're called residualizing labels. And labels that can diffuse through the cellular membranes, they are halogens like iodine, bromine, fluorine. They are able to diffuse outside, pass through the membrane, and leak outside of the cell. So it's quite a complicated concept to understand, but this is really explains the results. This really explains why we see what we see in our studies and with imaging. So how fast is internalization in cancer cells compared to internalization in normal organs? So if we think about cancer cells, we know that generally internalization is quite slow. It's not very, very fast. So if something targets the cancer cell, it usually stays on the surface of the cancer cell. But if someone is taken up by some cells in the liver or in the kidneys, the internalization in normal organs such as liver or kidneys or some other organs is very fast. So the label is quickly disappearing from the cell surface and then can be diffused. So if we have slow internalization in the tumor but fast internalization in normal organs, that means that we can use non-residualizing labels. We can use iodine, for example, because with iodine, the label would just stay on the cell surface of cancer cells, but it would be diffused in normal organs. And this is exactly what we see on these two pictures. So it's the same protein, but labeled with two different labels. On the right side, we have non-residualizing label or halogen label. On the left side, we have residualizing label. It's a kind of metal-like properties. So the protein is taken up in gastrointestinal tract and in liver, so we see a lot of activity here. It's also taken up in tumor, as we can see here. But in case of residualizing label, because it's taken up by all cells in the body, degraded, and the label cannot diffuse outside of the cell, so it just stays there. We see a lot of activity in normal organs. But when we talk about the non-residualizing labels, the halogen, as I said before, internalization in normal organs is very fast, which means that the label is basically, the protein is taken up by normal organs and the label just leaks out from normal organs. So we don't see so much activity in normal organs, but in tumor, internalization is very slow. So it just stays on the cell surface, it doesn't go anywhere, and we still can detect quite a high signal from the tumor. So I just want to ask you if you think this concept was complicated and if you understood something.
I have a question.
Yeah.
On the previous slide before this one.
Yeah.
So if I understood it correctly, both fast diffusion and slow externalization will give us signals.
Okay, once again.
That's what I lost you on the part where they get into make cytoplasm.
Yeah.
And then you mentioned that since it's acidic pH, the proteins will be destroyed and will move on to the next level. Now, from there, the fast diffusion one will give us a signal, and the slow externalization, will it not give a signal? Will this one also give a signal? This is where I lost you, basically. This is where I didn't really understand.
Okay, so if we just talk about cancer cells,
Yeah.
and we have a label on the cell surface that goes through this process very fast and diffuses out, we lose it. So for radiohalogen, for non-residualizing labels, if internalization is fast in the tumor, it's not good. If we know that our protein of interest, a monoclonal antibody, internalizes very fast in the tumor, we should not use non-residualizing labels. We can only use residualizing labels because we just lose all our signal from the tumor. But if we have fast internalization, then we should use radiometals because that would allow us to still have the signal inside. Doesn't matter if it's on the cell surface membrane or inside the cell, it's still in the tumor. Okay. So for every protein of interest and for every target, we study this process in the lab. We study how fast our protein of interest together with the receptor, how fast they internalize in cancer cells. And for example, if we see that after several hours, all of the protein has disappeared from cell surface and went inside the cell, and then it degraded, and then the radio label, if it's iodine, it just leaks out of the cell. So if we see that this is happening, we don't use iodine as a good candidate. We don't consider iodine as a good candidate for further translation. That means that this kind of protein should be labeled with a metal.
Okay, great.
Yeah, so this is something that we study in the lab. Yeah. So once again, what we see here is that we see if we see in this kind of picture good retention in the tumor, that means for both non-residualizing and residualizing labels, that means internalization in tumor is not so fast because we still can see it. But internalization in normal organs is very fast, and in case of collagen, we just lose it. In case of metal, it's still there. So actually, sometimes we use two labels and just to confirm the same process, basically. So we can go a bit further. So basically, we have two categories of the label: non-residualizing label or residualizing label, and we have various half-lives that would determine the optimal imaging time from few hours to several days post-injection. And then comes another level of complexity when we talk about the labeling chemistry itself. There are only certain methods that you can use to attach your radionuclide to the targeting molecule. And for carbon-11, it's usually through amines on the proteins, amino group on the proteins, through fluorine, for fluorine 18, some prosthetic groups or aluminum fluoride. That's what Bo will talk about in his lecture. For bromine and for iodine, it's a halogen chemistry. And for the radiometals, it's a chelation by chelators. So a little bit about the labeling chemistry. I'm sure you know that labeling of proteins can be done by attachment to the side chains of amino acids. I'm sure you know the structure of amino acid, but just to remind that it consists of an amino group, of a carboxy group, and of a side chain. So the peptide bond is a bond between carboxy group and amino group. So the CO and H is a peptide bond, but what you can see in here in yellow are the functional groups, the groups that are different for all amino acids. That's how the amino acids differ from each other. They differ by side chains. And depending on the property of the side chain of the functional group, we can use different kind of chemistry. So if we have a cysteine in our protein, we can use a group that would react with the style group. We can use a label that would be attached to this style group. And if we have just one cysteine per molecule, usually in proteins, cysteines are not very common, and sometimes you can introduce a single cysteine by genetic engineering. It's very convenient because if you have a single cysteine, you only have one potential reaction site with your label, so you can only attach one label per molecule. And it's a very precise modification. It's good because you know exactly the site of modification in your biomolecule, and you know the number of labels you attach per molecule. And what is the opposite of cysteine? It can be situation with lysines. So lysines are amino acids with the free amino groups. And usually lysines are very common in proteins. For example, in monoclonal antibodies, there are really a lot of lysines, around maybe 70 lysines or more that can be used for modification. So, of course, if we use an amine-directed chemistry, a chemistry that reacts with amines, we lose control over how many labels would actually be attached to the molecule. It's called non-specific or random labeling, and less control over modification. So I just want to ask you if you have heard about antibody drug conjugates before.
Kind of rings bells, but not in detail.
Okay, so basically the antibody drug conjugates are conjugates of antibodies and some cytotoxic drugs that are attached to each other, and usually they use non-specific or random labeling, and which means that the number of drug molecules for each antibody can really vary a lot in a single dose that you give to a patient. And that has been really the major problem, because if you don't know how much drug you give, you don't know what effect you will get in a patient, and the side effects and so on. So from the regulatory point of view, like the FDA, Food and Drug Administration in US, really has quite strict rules about that, and sometimes you can even reject those candidates. So basically the site-specific modification is better than a non-specific modification, but sometimes you're limited by the chemistry you can use. So sometimes you have to use the amino-reactive chemistry. And now we can talk about the attachment of radionuclide to our biomolecule. So we have to use chelators for attachment of metals. Do you remember, maybe from your pharmacy studies, do you remember about chelation? Like, have you heard at all about this, or it's totally new?
I've heard about it, but I've forgotten the details again. Yeah.
Okay, so chelation is a process where we have our metal ion, and a metal ion forms it basically can be held together by a chelator around it, and can form some ionic bond. This way the metal can stay inside the chelator pocket. Because the metal is just an ion, it cannot be attached directly to the molecule. We have to first attach it to the chelator, and then attach this complex of chelator metal to the biomolecule of interest. So that's why we use chelators as this kind of intermediates to attach the radiometals to the biomolecule of interest. And basically, metal chelation is a process of formation of coordinate bonds between a ligand. It's called a polydentate or multiple bonded ligand because it forms several bonds with the metal atom. So some other names that you will see for chelators can be called chelators, chelants, or chelating agents. It's all the same. And very common chelator that you see on all food packaging, in most of the food products, in pharmacy, in cosmetics, is called EDTA. So it's ethylene diamine. So it's an ethylene bridge here. We have two amines, diamine, and then we have tetraacetic acid. So we have 4 residues of acetic acid, 4 acetate groups. So a short abbreviation for this is EDTA. And it's actually used in if there's a heavy metal poisoning, it can be given IV to chelate these heavy metals, remove them from the body. And what we use in the lab, what we use for radiolabeling is an analog of EDTA, is called DTPA. So it's a similar structure. We have these 2 ethylene bridges, so it's a diethylene. Then we have three amino groups, 3 amine, pentaacetic acid, because we have 5 acetates. So DTPA is a very common chelator for radiometals. And you see it's called acyclic chelator. So there are cyclic chelators that form a cycle, and this is like just a linear structure, so it's acyclic chelator. So these are the examples of acyclic chelators. So we have our DTPA here that in this picture you really see the structure like a 3D structure of the complex between a radiometal in the center and then the coordinate bonds that are formed between oxygen and between nitrogens in this chelator. And yeah, you can see that in the formation of this bond, there are also nitrogens that participate and oxygens as well. So if the charge of the metal is 3+, that means that for its ideal coordination, if there are 3 oxygens with a charge of -, then the overall charge of the complex will be neutral, will be 0. So if the charge of the metal is 3+, and we have 3 free oxygens minus that can form a complex, then it's a neutral complex. But then there are also nitrogens that can participate in the formation of these coordinate bonds, but they don't add any charge, additional charge. And so we have discussed the structure of the chelator itself. Then we need to have some kind of way to attach this chelator to the biomolecule. And we need to introduce another functional group that can react with these reactive groups on the protein surface, that can react with amino groups or with the cysteine groups, something that can be attached to the protein. So sometimes you can see like this term called bifunctional chelators. So the chelator can have 2 functions. First function is the chelation of the metal, and the second is attachment to the protein. And on all of the structures, you can see the same DTPA chelator, but you can see various functional groups that are introduced into the structure to be able, in order to afford different kind of labeling chemistry, different kind of reactions. So you can, when you want to buy a chelator commercially from some company, you can usually find all kind of modifications of the same chelators that would react with different functional groups in your protein. And now we can just look at a second category, or chelators. They are called cyclic chelators. They are called in this way because they form a microcycle. So the nitrogens in the structures, they form, they all connected through the ethylene bridges in a big cycle. And then the atom of the metal comes in the center of the cycle. And then usually there are carboxy groups. Some of them are used for chelation itself. They participate in the formation of the coordinate bond. And usually one of these carboxyl groups is used for attachment to the protein. So if it's 4 nitrogens, it's DOTA. If it's 3 nitrogens, it's NOTA. And there are all these kind of derivatives of the chelators. And then we can also have various modifications. So if some specific radionuclide requires more stable complexation, some of these alternative chelators can provide that extra stability of the chelation. So it's really should be for developed or selected for each nuclide individually sometimes. And here you can just see examples of possible modifications of chelators, but you already know the general principle that the metal is chelated by nitrogens and carboxy groups that form the coordination complex. And right now we will go into some examples of radiometals that we use for imaging, and we will look at different chelators that can be used for chelation. And so again, you just need to know a general principle. You don't need to remember each individual molecule. It's just for you to understand how it works in general. So we can look, for example, at yttrium 86. It's a PET. It's used for PET imaging. And for this particular nuclide, if it's complex, it can form complexes with DOTA, with this chelator. It can form stable complexes, but in order for yttrium to go inside the cycle and to stay there very stably, you need to heat it for a long time, for high temperatures, for long incubation time. So if you have a biological molecule that is very sensitive to high temperature, for example, antibodies, most monoclonal antibodies cannot be heated higher than 40 degrees or 42 degrees. So it means that you cannot use this kind of chelator with the metal. If you want to label your monoclonal antibody, it would just degrade and lose the function. So something that we need to consider is conditions of our labeling, what temperatures, what time we use. So this chelator is not very optimal, but there are some other chelators for yttrium 96 that are more optimal. For example, these acyclic chelators such as DTPA. And the second nuclide that we can consider is zirconium 89. So zirconium 89 is extremely popular for labeling of monoclonal antibodies because it has long half-life, and monoclonal antibodies have a long half-life. And the chelator that is used for labeling with zirconium has this quite a complicated structure and name, and shortly we call it DFO. So DFO is a chelator for zirconium, but it's not an ideal chelator because it doesn't hold zirconium very well, and when it's injected in the body, zirconium diffuses out of the chelator into the blood, and then it can go into the bone. So you can see unspecific signal from the bone uptake. So the good things about labeling with zirconium using DFO chelator, it's a relatively mild conditions, so good for monoclonal antibodies, but the disadvantages is that it's not very stable in blood circulation, and yeah, for sometimes they can be released in the bone and liver. So in this example, you can see imaging of a patient with the HER2 expressing metastasis, and using trastuzumab is a monoclonal antibody that targets HER2 receptor that was labeled with zirconium-89 with the chelator DFO. And this is images 5 days post-injection. And what we can see in this patient, we have a lot of activity still in arteries. That means, or we can see contrast in the arteries, which means we have a lot of activity in the blood, which means that even 5 days after injection, our monoclonal antibody is still circulating, and it just gives us a lot of background. So you see this blurred gray is basically your background level. So because trastuzumab has a very long half-life, it's not ideal for imaging, but it's something that people like to use because it's convenient. And we can see some individual metastases, for example, here in vertebra, but it's not, or in liver, not visualized so well, but we can also see uptake in the bones. And this uptake in the bones can really indicate instability. And sometimes you cannot distinguish, is it uptake in the bone or is it the bone metastasis? So from this point of view, zirconium DFO is not a very good chelator. And here I just want to say about antibodies, about what we do in our lab. We work with small targeting probes, so monoclonal antibodies have large size, they circulate in blood for a long time, and the imaging can only be done days after injection, and still it's not good. And it can also give you false positive results, and these molecules are very sensitive to temperature and pH. But we work with antibodies which have a small size, they have super fast kinetics, they excreted from the blood in minutes or in hours, so we can do imaging with a very high contrast just hours after injection. And they're also very robust. They usually tolerate high heating, which means that we can use a lot of different labeling methods and not worry that we will destroy our targeting protein. So this is just a comparison between a patient that has HER2 expressing metastasis, and on this case, 5 days after injection, we still cannot clearly see the tumors because everything is just gray, and we have a lot of activity in the blood, in the arteries, which contributes to background. But when we have antibody, which is a very small tracer, it's excreted from blood very fast, so we have a very high contrast. We can see really on this patient individual metastasis in the bones, so it's a very late stage cancer. We see a lot of metastasis all over the body with a really, really high contrast. So that's how the size of the biomolecule can affect your final image. So we still have about 10 or 15 minutes of time, and now we will quickly go through various different examples of different chelators and properties and images that we get finally. So what we do in the lab, usually we study which chelator would work best for every radiometal. So if we talk about gallium-68, it has a charge of 3+ on its own. And then if it's complex bimetal, depending on the number of these negative charges on oxygens, depending on the number of free carboxyl groups, the total charge of gallium and the chelator can be either +1, 0, 0, or -1. So in this case, we can take 4 different chelators, and they would all have different charges. Why it is important? Because eventually it affects the uptake in normal organs quite a lot. So we know from experience that if a charge is positive, it's usually not so good because there might be some uptake in liver. In healthy organs, we see background, but if charge is negative, generally it's better because we see much less background. And another thing that can also be affected is affinity to a target. This is just a quick example of a single molecule called RM26 that was labeled with gallium, but different chelators. And in case of the TAGA, which had charge -1, the affinity was shifted a little bit because of the charge and the conformation of the metal chelator complex. So that can affect binding to the target. For cobalt 55, it's another positron emitting nuclide, and for example, one site of production is in Denmark. Yes, and it has a charge of 2+. And depending on the chelator, there can be complexes of different charges. So for cobalt, it's actually quite nice because all of them have a negative charge, and negative charge is good because we have more hydrophilicity and less uptake in normal organs and less background. And a cobalt doesn't have a preferable chelator, so many of the chelators work for cobalt. Also, another thing that I forgot to mention is maybe it's a bit too much details, but I just want to say that gallium actually works better for NOTA and NODAGA for three cycle chelators, triaza chelators as they are called. It works better for triaza chelators because gallium has a small ionic radius and the cavity of the chelator is small as well. So it's a very, very good fit. Sometimes we have to look at the ionic radius of our metal and the fit of the metal chelator to determine which complex will be the most stable. So once again, we can look at another example where we have the same peptide. It's a bombazine antagonist, so it targets bombazine receptors. And here we have a labeling with cobalt 55 or with gallium 68. And it's the same molecule, but we see a different level of signal in normal organs. So in the case of, in both cases, we see some uptake in normal organs as well as it is in the tumor, but it looks slightly different from them because of the different properties of the chelator metal complex. And we can see it once again on this slide. For example, in case of gallium, we see some residual signal in normal organs such as gastrointestinal tract or kidneys, but we don't see that in case of cobalt 55. We only see the signal in the tumor because of the really this minor differences. You think it's such a minor difference it's just the nuclide itself. How can it affect so much? But actually, it's the structure of the peptide is small on itself, very small. So even minor modification can really introduce big changes when it comes to the biological behavior. And just another example is an anti-HER1 Fibody molecule that was labeled with 2 different nuclides, and again we see quite a different picture. So it's the same molecule but 2 different nuclides. And if we look at the so-called biodistribution graph, which describes signal in different organs with different doses, we can really see that with indium, which is was on the left side, we see like a huge bulb in the center. So the uptake activity in normal organs is quite high in liver, high in GI tract, high in carcass. But when it comes to cobalt, the activity is much, much less in normal organs. So that's why we don't see so much activity accumulation in normal organs. So activity accumulation in normal organs is something that we call non-specific uptake. So we try to minimize that as much as possible. So we only want to see uptake in the tumor in the ideal case. But sometimes we have to work on the properties of our biomolecules, do this kind of optimization. For example, if we started working with indium, we see that it's not good, then we can do some optimization, try cobalt 57, and then we can really see that it improved. So for every biomolecule, ideally, we can find a suitable method of labeling, suitable radionuclide. So these are just some examples. Scandium-44 is a very rare nuclide, so I don't want to spend too much on it, just so you know that it can also be chelated by the common chelators. And there have been some studies that showed the potential of using scandium-44 for imaging, but scandium is rare. It's only available in 2 places in the world. It's not so widely used. So gallium-68 is much more widely used compared to scandium, but their chemistries are similar, the labeling chemistries. And we have also, there were some studies from our group that used scandium-44 for labeling of this HER2 targeting antibodies and imaging of cancer in mice. Another type of chemistry is copper directed chelators for copper. So the commonly used chelators are not very useful for copper, so they have to be there were some studies where they compared copper that was chelated by NOTA and copper that was chelated by DOTA and attached to the pombuzine analogs. And then they could see that for the DOTA, there was much higher uptake in normal organs, so it was not very optimal. So what we see again is that we see not so much in the tumor, because everything just taken up by liver and doesn't reach the tumor, so we don't really see good visualization of tumors. And then there were alternative chelators that were developed for copper that suited much better compared to NOTA. They are called like TETA or T2A. And in our lab, we have studied previously both NOTA and NODAGA for imaging with copper, and we could see accumulation in the tumor, but we also saw some accumulation in normal organs in the liver, for example. So then the second study after that that was done recently have really introduced some modifications, so introduced negatively charged residues of glutamates. They have negative charge, so if you remember, when we increase the negative charge, we increase the hydrophilicity, so we decrease uptake in normal organs. And if we look at our uptake in the liver, we can really see that originally it was around 5%. With the second variant, with the G chelator was used as a control, the uptake increased, which is not so good, but with our modification, with the hydrophilic modification, with glutamates, the uptake really was suppressed. So that was like the optimal finding for this kind of molecule. And here we can really see the images from the slide before. So they have used NODAGA, which was compared as a comparison, which was best from previous studies, but it has some liver uptake. Then there is a second chelator, T2A, but it also gives some liver uptake, but with the T2A and the triple E with glutamates, we have negative charge, so we have really the lowest uptake in the liver and normal tissues and the highest uptake in the tumor. So the tumor is really bright compared to this one, for example. So that was like the optimal chelator for copper. And then we can also consider indium, though indium is actually not used for PET, but it's used for SPECT. But just to show that for indium we can use similar types of chelators, and we can also look at, depending on the type of the chelator, we see a totally different picture, which is quite fascinating. It's just a small modification in the structure of the molecule, but it really affects the result that we get. So in these three cases, we see good tumor targeting. In the case of Nadaga, we don't even see the tumor targeting. But because everything goes in the taken up by liver, it goes into the bile, then from the bile it goes into the intestines, and what we see is uptake in the gastrointestinal region. So when we minimize the uptake, minimize the unspecific uptake in normal organs such as liver, we can really minimize background and much increase the contrast of imaging and the specificity of imaging. So for example, if a patient with cancer will have some metastasis in the liver or in the intraperitoneum region, and we want to visualize this metastasis, we should not take this molecule because we will not be able to distinguish where is our specific signal, where is metastasis, where is the tumor. We will not be able to distinguish it from the unspecific signal. But in this case, it's a good candidate because it's very specific to the tumor. We don't see unspecific signal from normal organs. And yeah, here is just another comparison how we can really optimize the properties and get, if originally the imaging was not so good, we can do some optimization and improve the quality of the image. And another thing is optimal labeling conditions, just another example from the lab. We should also remember what are the conditions that we use, and if we use, for example, 60 degrees, we heat the molecule when we do the radiolabeling, we have to heat at high temperature in order for the metal to be held by the daughter for the chelation to happen. So we need to heat it, and if we heat it at 60 degrees, the biological molecule preserves its functional properties, so it can go to the tumor and bind to the tumor, and we see targeting. But if we do the radiolabeling at 85 degrees, in this case, this molecule doesn't tolerate it, and what we see is we just see unspecific uptake. So the biological properties of the molecule were destroyed. That means that everything that is injected intravenously is just taken up by liver and further on processed by the liver and kidneys and spleen. So it's something that we don't want to see, definitely. So we need to find optimal labeling conditions. So sometimes we know it from experience, what works for every kind of molecule. So really the take-home messages from this lecture could be that our choice of labeling method, our choice of nuclide and chelator depends on biological half-life of targeting molecule. It depends on cellular processing and internalization speed. If it's internalized fast in the tumor, then non-residualizing halogen label is not good, then it's better to use radiometal. But if it's not internalized very fast in cancer cells, then we can use both radiometal and radiohalogen, both residualizing and non-residualizing label. We also need to think about the preservation of biological function of the molecule. We should take into account binding site composition, amino acid composition of the protein, which amino acids are in the protein, which labeling methods we can use for attachment of the label to the protein, and we should consider the profile of our biomolecule. We should not make it too lipophilic and introduce as many negative charges and as much hydrophilicity as we can to the protein. Yes, so that was all that I wanted to say for this lecture. Do you have any questions? Something that was not clear?
So I have a question. So just to confirm with you, you mentioned that if there is a negative charge in the nuclide or metal in the metal,
Yeah.
It increases the hydrophilicity, and therefore it gives us a better background, and also the fact that it does not penetrate.
Yeah, so it the negative charge gives us lower background because there is no uptake in normal organs and tissues, or there is less uptake in normal organs and tissues. So less uptake in liver, generally less uptake in gastrointestinal tract. That's why negative charges are better. I know that the next lecture, Bo should also log in at about this time. We agreed that he will just join this Zoom meeting. He said he would log in at about 14:45, but he didn't log in, so I will just send him an email that he can log in. In the meantime, you maybe you will have some other questions that you want to discuss. So I know that this lecture can be quite complicated because we are looking at a lot of different parameters at the same time. We are looking at physicochemical properties of the nuclide in respect to the radioactive decay, in respect to the radiation types that are produced, images that will be resulted from that. And at the same time, we are looking at the labeling chemistry, the way that our radio label can be attached to the molecule of interest. We're also looking at the biological properties of the molecule, that it's still able to be functional. We evaluate it in cell-based assays. We look at the biodistribution in living systems, in living animals. We look at the tumor targeting. Yeah, so it can be quite a lot of information to process. So I understand that it can be quite difficult.